But why I'm not getting the insulin protein? Post-transcriptional gene silencing. Who is the guy who is responsible? Protein is not produced. Where? How do you understand this? Protein is developed, insulin is there. The insulin is not functioning. Why? Post-translational modification of the insulin. The two chains is not combined together. It is a problem. Insulin is there, gene is expressed, protein is there, but it is not functional. Why? It's a connecting cysteine molecule is not connected, it's separated. It is not no more functional. That can also be addressed in the proteomics. Yes, this is one question comes here. Not all the plant pod does contain all the compounds. If you want morphine, this morphine will be abundantly present only in the latex of poppy. It is not in stem, it is not in the root, it is not in the some other part, but latex does contain lot. That is very precise manner compartmentalized. Okay, so I don't like botany. I am very strong synthetic chemist, medicinal chemist. I can do wonders. Whatever the geometric modification you want, you tell me, I will do it. Where is the problem? The problem is chirality. Synthetic chemist fails in chirality. Chi. This is a chi. Chirality of the carbon arrangement. You cannot beat the nature. Only can be done through the plant metabolism. Sir, I am super in medicinal chemist. I can synthesize whatever you want. Yes, you cannot. You cannot beat the nature. Chirality of the carbon valence is 4, the 4 and 4 chi. Different levels of organization, stereoisomerism. You cannot satisfy this only through plant. That is why till now it's a viable for the medicinal plants. Till today is the viable. The recent failure of chirality of carbon or chirality of other molecule cannot be achieved through synthetic medicinal chemist. That is the major drawback. So with that, this is the global scenario. Active pharmaceutical molecule comes from the plant, contribute how much money worldwide. So yes, of course, I'm a little bit touching with the molecule which is responsible, which I'm going to explain is neurofunctional molecule. So this is the Nodostachys jadamansi, jadamuni. It's grown in Himalayan region, and we have taken, this is a methodology we have adopted with the two final product, and we have just started for in vitro studies, but we have not done for clinical trials. We have just in vitro complete methodology what this, and we have used for high-end machine for post-translational modification and metabolomics study. This is the ideal machine, HT ESI QTOF mass spectrometry, and with that you can point, for example, my weight is 76.0341. 0.4 digit can measure the molecule. That is the resolution of that particular machine. So that is why post-translational modification can be studied. Epigenetic modification of the protein associated with the DNA, cytosine methylation in which location is get methylated, and DNA associated protein lysine acetylation can also be studied. You can locate and you can study what is the difference. Phosphorylation also can be studied. Summylation also can be studied. Epigenetic modification with this machine. So we have just isolated the compound, different pattern we have optimized and extracted, and we have profiled with that machine, and we got major peaks, 4 peaks, 1, 2, 3, 4. This 4 peaks with the ISO solution mass spec, we have identified what are the compound as it, and they are the compound. They are all is totally 7 alkaloid we have identified, 7 to 8 alkaloids. Among the 8 alkaloids, 4 of them are abundant based on the amount. abundancy, and based on this abundancy, we have start purify with the more abundant alkaloid, 2 alkaloid, actinidium, glaciovine, 2 alkaloid. So this 2 alkaloid, yes, we used several chromatographic technique. Also, we have used for HPLC for fractionation, and we got the purity. And this is the purity of HPLC purity and percentage, 98 and 94 percentage both of the alkaloid. And again, we have confirmed whether we are moving towards the correct compound or wrong compound. Again, fragmentation pattern of that particular alkaloid have been done with the mass value, and we have confirmed, yes, we have isolated actinidium and we have isolated glaciovine. Once confirmed, this is the first report, we have isolated this 4 alkaloid and we have reported 2 alkaloid purified and characterized. And again, we confirmed with the GCMS for, yes, this is the alkaloid or not. Still, we had doubt, and again, NIST library, we have injected and compared with the NIST library, yes, we got that probability percentage 92% of both the compound. Yes, this is okay. I don't want to go again and again Himalaya to collect the sample and to isolate the alkaloid which I want. What is the alternate technology do I have? Tissue culture technology we have adopted. Plant tissue culture, this is the methodology we have adopted for in vitro, and we have just teasing. If I want to make to act, I can ask the question, teasing you become ground state to excited state, you start react. The same way, if I tease the plant by using a biotic and abiotic elicitor, I am simulating, I am giving problem to the cell. While giving the problem to the cell, plant start respond against that molecule, that after responding, whether this response could be my compound of interest alkaloid or not. Yes, we have used several biotic and abiotic elicitors. Lipopolysaccharide is a bacterial derived molecule, and salicylic acid, again, it's a plant hormone. Now we are using methyl salicylate, acetyl salicylate as a paracetamol active ingredient, methyl jasmonate, chitin, the exoskeleton of the fungi, the insect, and except zygomycetes fungi, rest other group of fungi, cell wall is made up of chitin, and we have used different concentration, and this is the behavior of the cells after treatment of different concentration of the biotic and abiotic stress factor used. And we have quantified again actinidium and glioisin. Yeah, this is, if you see that chitin treatment, it enhances, and lipopolysaccharide treatment, it enhances 12.64 glaciovine, and like chitin treatment and salicylic acid treatment is 6.74 get fold increases. Salicylic acid for better accumulation of actinidium, chitin is the better for glaciovine. Again, we have confirmed through HPLC and other technology. Again, we went for antioxidant scavenging potential of this alkaloid by analyzing DPPH, ABTS, and FRAP. And again, we take that the cell and treat it with the cell line, and we have observed how the neuronal cell lines behaves. And yes, of course, this is yesterday that one of the professor, he gave the talk, he also interested, I want to do the molecular docking against the neuronal cells targets. You tell me the targets and you give the compound and I will do that. And he has selected and he did the Professor Velmurugan. This is the molecular studies for against the actinidium and glaciovine. And these are all the targets, protein which have been selected from the neuronal cells. This is a summary of jetamancy. The another story is the Camptothecin. Nadostachys jetamancy 1 and Nathapodiaetes pneumoniana. The plant grows in the Karnataka to Vindhya Satpura hills, that area. This is the mechanism which I have said, inhibitor for unwinding of DNA, complementary DNA. This is This is a plant. And again, we have adopted, we have quantified the amount of camptothecin present in the bark, stem, root, or sap. This three part we have, and in the bark only we obtained good amount of camptothecin. And again, we have confirmed whether we went for camptothecin or not. Again, one more quite interesting, the camptothecin is methylated. Methyl group is added into the camptothecin, and we got the two peaks, and it made us to, it is a impurities or something